Welcome to the Deep Dive, the place where we strip away the jargon and turn complex cellular machinery into surprising, accessible knowledge.
Today, we are undertaking a meticulous, step-by-step deep dive into, well, the foundational energy capture process on Earth, photosynthesis. We are exploring the chloroplast, focusing on every major idea, mechanism, experiment, and adaptation detailed in the relevant chapter summary.
Our mission is to really understand how plants pull off what seems impossible, converting simple CO2 and water into the complex organic structures that sustain, well, the entire global food chain. It's the engine of life, and we're going to open the hood and look at every single piston and valve.
To really set the stage, we have to go back, way back. We have to remember the context of early evolution. The first forms of life were what we call heterotrophs.
Meaning they were basically grazers.
Exactly. They were dependent on preformed organic compounds that were just, spontaneously generated and dissolved in the primeval oceans. They had to rely entirely on existing resources.
And I imagine those resources were pretty finite. As life started to get more abundant, that supply of abiotic organic stuff just couldn't keep up.
It couldn't. And that scarcity, that pressure, drove what is probably the greatest metabolic breakthrough in the history of life.
The rise of autotrophs.
The rise of autotrophs. These are the organisms, like plants, algae, and some bacteria, that are capable of manufacturing their own organic molecules from really basic inorganic components, mostly CO2.
And we can split them into two main groups, right, based on where they get their energy.
We can. You have chemoautotrophs, which use chemical energy stored in inorganic molecules. But our focus today, the real powerhouse that fuels the vast majority of ecosystems, is the photoautotrophs, which use radiant energy from the sun.
Okay, so let's unpack the evolutionary difficulty here, because the first photoautotrophs weren't nearly as powerful as, say, a modern plant. They weren't using water yet.
That's a critical point. No, they weren't. The early pioneers likely used reduced inorganic compounds, like hydrogen sulfide, H2S, as their electron donor.
So their version of photosynthesis would have been CO2 plus H2S.
And that would produce sugar, water, and pure elemental sulfur.
And we can still see relics of that ancient pathway today, right?
Yeah.
In places like sulfur springs, where you have these symbiotic green sulfur bacteria.
Correct. They're still doing it. But here's the pivotal moment in planetary history. Hydrogen sulfide, well, it's just not abundant or widespread enough to support a global ecosystem. The real game changer, the event that flooded the atmosphere with oxygen and made complex life possible, that happened around 2.7 to 2.4 billion years ago.
And that was the appearance of cyanobacteria.
Cyanobacteria. They were the first organisms capable of performing what we call oxygenic photosynthesis. They switched the electron donor from H2S to the ubiquitously available, highly stable water molecule H2O.
And that changes the output completely. Now, combining CO2 with water gives you sugar plus molecular oxygen, O2.
So why was this switch so incredibly difficult? Well, it comes down to chemistry. Water holds onto its electrons far, far tighter than hydrogen sulfide does. We can actually quantify this thermodynamically. The difference in redox potential is huge, about 1.07 volts.
That sounds like a technical number, but I mean, what does that translate to for a single-celled organism?
It translates to a monumental engineering challenge. It demanded the evolution of a completely new set of machinery. Machinery capable of generating an oxidizing agent so incredibly powerful that it could literally strip tightly bound electrons right off the oxygen atom in a water molecule.
So it was like choosing the hardest possible route, thermodynamically speaking.
It was, simply because water was everywhere. That evolutionary pressure is the reason the photosystem machinery we're about to discuss is so powerful and so complex.
Which brings us directly to the organelle that houses all this machinery, the chloroplast. We're essentially talking about a tiny, captured, ancient power plant.
That's the core of the endosymbiosis theory. The idea that a primitive eukaryotic cell engulfed one of these newly evolved oxygen-producing cyanobacteria, and over millions of years, that bacterium lost its ability to live on its own, and the relationship became permanent. The chloroplast became a part of the plant cell.
And the evidence for that origin story is still, written in the organelle's DNA.
Oh, absolutely. Modern chloroplasts are genetic mosaics. They still have a small, circular, double-stranded DNA molecule and their own prokaryotic-like ribosomes. It's a clear echo of their cyanobacterial ancestry.
But the plant cell is in charge now.
It is. The plant's nuclear genome has basically assimilated control. The vast majority of chloroplast polypeptides, we're talking up to 3,500 of them, are now encoded by nuclear DNA, synthesized out in the cytosol, and then imported back into the organelle.
Okay, so with that evolutionary history in mind, let's move inside the plant leaf and
Yeah.
really examine the architecture of this engine, focusing on the structures in section 6.1.
So chloroplasts are found mainly in the mesophyll cells of the leaves. If you picture a cross-section of a leaf, you've got these tightly packed palisade cells right near the surface.
And they're just crammed with chloroplasts. It's all about maximizing light capture.
Exactly. And their placement close to the surface highlights that tight link between needing to absorb light and needing to let CO2 in through the stomata.
The physical size and the sheer number of them is just astonishing to me. They're lens-shaped, anywhere from 2 to 10 micrometers long. Each one is as large as an entire mammalian red blood cell. And they divide on their own.
They do. They arise through binary fission from these precursors called proplastids, which is very much like their bacterial ancestors would have divided. The chapter even shows a micrograph of an Arabidopsis mutant where you can see this asymmetric fission happening. It's a direct visual link to their past.
We should probably mention the historical context here, that elegant experiment by T. Engelmann back in 1881.
A truly beautiful experiment. He was looking at Spirogyra, which is a filamentous green alga under a microscope, and he introduced aerobic bacteria into the water.
Bacteria that need oxygen to survive.
Right. And he saw that the bacteria would actively swim and just congregate. They would cluster only near the chloroplasts, using the tiny amounts of oxygen being released. And that proved, beyond a doubt, that the chloroplast was the precise site of photosynthesis.
Okay, so let's dive into the core structure, starting with that envelope. It's a double membrane, just like the mitochondria.
It is. The outer membrane has these things called porins, so it allows some small solutes to pass through, though it is somewhat selective. The inner membrane, however, is a different story. It's highly impermeable.
The real gatekeeper.
Exactly. Any necessary metabolite or ion has to pass through specialized transporters embedded within it.
But the real action, the real complexity, is in that internal membrane system, the scaffolding that holds all the machinery.
That internal system is organized into these flattened, interconnected sacs called thylakoids. These sacs contain every single component needed for the light-dependent reactions. And when these thylakoids are stacked up neatly, like a roll of coins, we call those stacks grana.
And the geography inside the chloroplast is defined by two key fluid-filled spaces, separated by those thylakoid membranes.
Precisely. The space inside the thylakoid sac is called the lumen. This is where protons, H+, are accumulated and concentrated during the light reactions, creating a very acidic environment.
And then outside the thylakoids.
The space outside the thylakoids, but still inside that outer envelope, is the stroma. And this is the alkaline, fluid-filled factory floor where the second stage of photosynthesis, actually building the carbohydrates, takes place.
You know, the book points out that the thylakoid membrane itself has a really unique chemical composition, which is critical for how it works. It's got a lot of protein, but very little phospholipid.
Yes, that's right. Instead of phospholipids, it's unusually rich in these galactose-containing glycolipids, like monogalactosyl deoxyglycerol. But the really important detail is the fatty acid composition of these lipids. They are highly unsaturated.
Meaning they have lots of double bonds.
Lots of double bonds. And what that does is it makes the thylakoid membrane exceptionally fluid.
And why is that specific lipid chemistry so essential for a process as fast and high energy as photosynthesis?
Because that fluidity is paramount. The whole system relies on the rapid movement and lateral diffusion of these huge protein complexes. The photosystems, the cytochrome bridge, the mobile electron carriers, they all need to constantly move around and interact across that membrane surface to keep the electron flow going.
Okay, with the structure established, let's transition into section 6.2 and get that big picture overview of the metabolism. We need to really nail down that it's the oxidation of water that we touched on earlier.
This takes us back to the 1930s. And a hypothesis from C.B. Van Niel. And it really flew in the face of the prevailing wisdom at the time, which was that light was splitting CO2.
Right.
Van Niel suggested a general reaction, he said. CO2 plus some electron donor, which he called H2A, yields sugar, water, and the oxidized residue, A. And when you apply this to plants where H2A is water, it means that the oxygen released must come from the oxidation of water.
And this was finally proven, experimentally, in a classic 1941 experiment by Rubin and Common using a heavy oxygen isotope, O18. Can you walk us through those results?
Sure. They ran two parallel experiments on green algae. In the first one, they gave the algae labeled CO2 carbon dioxide with that heavy oxygen and just normal unlabeled water.
And when they collected the gas that came out?
The oxygen was completely unlabeled, normal oxygen. This immediately ruled out that old idea that CO2 was being split.
But it was the reverse test that was the real nail in the coffin?
Precisely. In the second experiment, they supplied unlabeled CO2, but this time they used labeled water, H2880, and the resulting oxygen gas they collected was entirely made of that heavy isotope, 1802.
Yeah.
It was the definitive aha moment. The oxygen released during photosynthesis is derived exclusively from the water molecule, not from the CO2.
So on a really fundamental level, we can just think of photosynthesis as the complete reversal of cellular respiration, just powered by light. Respiration reduces oxygen to water and releases energy.
And photosynthesis oxidizes water back to oxygen, which requires a tremendous input of energy.
And that energy input drives the two major stages of the whole process.
Stage 1, the light-dependent reactions. This is where the light energy is actually captured and converted into useful chemical forms. The output of this stage, which all happens in the thylakoid membranes, is chemical energy stored as ATP and reducing power stored as NADPH.
And then stage 2, the light-independent reactions. People sometimes call them the dark reactions.
Right, and these take place in the stroma. They use to actually synthesize carbohydrates from CO2. And it's worth noting, while they don't require light directly, they operate much, much faster in the light because the enzymes involved are light regulated.
Before we really dive into the molecular nitty-gritty, let's just appreciate the scale of this one more time. We are talking about converting 500 trillion kilograms of CO2 into carbohydrate every single year.
It's incredible. I mean, this two-stage process defines the sheer scale of life on this planet.
Okay, incredible indeed. Let's transition into section 6.3 and 6.4 and really analyze how life captures light and channels that energy, the physics of it all.
So we think of light as waves, but biologically it's more useful to think of it as individual packets of energy. We call them photons or quanta. And the energy of a photon is always inversely proportional to its wavelength.
So shorter wavelength light, like blue light, carries more energy than longer wavelength red light.
Exactly. And when a photosynthetic pigment like chlorophyll absorbs a photon, that energy is used to boost one of its electrons from its normal ground state up to an outer, higher energy excited state. But this high energy state is incredibly fleeting. It only lasts about a billionth of a second.
And if you had that chlorophyll molecule just sitting in a test tube, that excited electron would immediately fall back down and it would release that excess energy as heat and a little flash of light. Fluorescence.
Right. But in the chloroplast, fluorescence is actually very faint. And the reason is that the plant is performing this ultra-efficient energy handoff. The excited electron is immediately transferred to an electron acceptor molecule that's embedded in the thylakoid membrane. It happens before the energy can dissipate.
And that rapid transfer is the initial harnessing of light energy.
That's the first step.
The principal player here, of course, is chlorophyll. Visually, we all know it appears green because it's strongly absorbing light in the blue and red parts of the spectrum, and it reflects the green wavelengths back to our eyes.
And if you look at the structure of a chlorophyll molecule, you can see it's perfectly designed for this job. It has this functional head called the porphyrin ring. This ring has a central magnesium ion, Mg2+ bare, which is different from the iron you'd find in a heme group.
And the ring is defined by that alternating system of single and double bonds, a conjugated system.
That's the key. That conjugated system delocalizes the electrons and allows the molecule to absorb visible light so efficiently.
And then anchoring that light-absorbing head into the thylakoid membrane is the second part, a long hydrophobic phytyl chain.
The chlorophylls are definitely the primary light absorbers, but plants want to maximize their catchment area, so they use accessory pigments, mostly the orange and red carotenoids.
Right, and these basically broaden the spectrum of usable light. They absorb energy in the blue and green regions where chlorophyll kind of misses out.
They do, and the carotenoids have a crucial dual function. It's not just about light collection, especially when the sun is really intense.
They also have a protective role.
A critical protective role. Their most important job is protection. When light intensity is extremely high, the whole photosynthetic system can get overwhelmed, and this can lead to the formation of these highly destructive excited states of oxygen, what we call singlet oxygen.
Nasty stuff.
Very nasty stuff. Carotenoids act like molecular pressure relief valves. They dissipate that excess absorbed energy safely as heat, which prevents the formation of these toxic radicals that could otherwise just destroy biological molecules.
And when you plot the overall efficiency of photosynthesis versus the wavelength of light, the action spectrum, it perfectly mirrors the combined absorption spectra of both the chlorophylls and the carotenoids. It confirms they're working as a team.
Exactly. Now let's move into the logistics. How is all this massive machinery organized? Back in 1932, an experiment by Emerson and Arnold using quick flashes of light revealed something pretty profound.
They found that it took roughly 2,500 chlorophyll molecules to be involved in the release of just one molecule of oxygen. And since we know it takes at least 8 photons for that reaction, it told them that not every single chlorophyll molecule could possibly be doing the actual chemistry.
And that insight gave us the concept of the photosynthetic unit. It's not a one-to-one ratio. Instead, you have hundreds of these pigment molecules acting together as a massive antenna system.
But only one special chlorophyll molecule within that whole unit, the reaction set of chlorophyll, actually performs the photochemistry. It's the only one that transfers its excited electron to an acceptor.
So the bulk of the pigments form the light harvesting antenna.
So how does the energy get from some random antenna pigment all the way to that single unique reaction center?
It's all based on physics. A bulk pigment molecule absorbs a photon, gets to its excited state, and then rapidly transfers that excitation energy from one pigment molecule to the next. And the rule is that the energy can only move to a pigment molecule that absorbs light of an equal or longer wavelength.
Which means a lower energy level. It's like a downhill path for the energy.
Exactly. The energy just cascades through the antenna, continually moving to lower and lower energy level molecules until it's finally trapped by the reaction center chlorophyll, which absorbs light at the longest wavelength of all its neighbors.
Okay, now for the master plan, the Z-scheme, which is laid out in figure 6.7. This involves combining 2 different power stations, 2 photosystems in a series. We touched on the thermodynamic reason earlier, but let's restate it.
Yeah, let's use an analogy. If you think of moving an electron from the extremely stable water molecule all the way up to the highly reducing NADP plus Phi, it's like climbing a massive energy cliff.
And a single photon is just a small booster engine.
Exactly. The total energy needed to make that entire climb is over 2 volts in the cellular context. But one red photon only delivers about 1.8 volts of energy. That single boost just isn't enough.
So the biological solution is a 2-stage rocket.
A 2-stage rocket. Perfect analogy. We use Photosystem II, PSII, to provide the first massive energy boost. It lifts the electron from the energy level of water to an intermediate level. Then, after a short coast downhill, Photosystem I, PSI, provides the second critical boost, raising the electron to a level far above what's needed to reduce NADP plus VAT.
And we should name those specific reaction centers.
Right. PSI uses P680, which is a chlorophyll dimer that absorbs light most strongly at 680 nanometers. PSI uses P700, another chlorophyll dimer, that absorbs light optimally at 700 nanometers.
The primary event in both of these systems is the same, though. Light absorption excites the reaction center, which then immediately transfers its electron to a primary electron acceptor.
And that instantaneous creation of a charge separation, a positive pigment and a negative acceptor, that is the fundamental conversion of light into chemical potential.
And that charge separation is what drives the electron flow across the 3 legs of the Z-scheme. From H2O to PSII, then from PSI to PSI, and finally from PSI to NADP plus I.
Exactly. So let's dive into the molecular mechanics of section 6.4, starting with PSII.
PSII is this massive integral complex. It's made of over 20 different polypeptides, and it starts by harvesting light using LHCII, light harvesting complex 2. And interestingly, the chapter points out that LHCII isn't always stationary. It can actually migrate along the thylakoid membrane and sometimes associate with PSI, which shows some dynamic resource management by the cell.
It's very dynamic, but when it's associated with PSII, the energy hits P680. The excited P680 transfers its electron to the primary acceptor, which is a molecule called pheophytin. It's basically a chlorophyll molecule that's had its central magnesium ion replaced by 2 protons.
And this is the first charge separation. We get P680 plus tau, which is a super strong oxidizer, and pheo, the reducing agent.
And to stabilize the system, that pheo immediately passes the electron on to 2 sequential plastoquinone molecules, PQA and then PQB.
And this reduction of that second plastoquinone, PQB, is a 2-step process. It needs a second photon and a second electron.
Right. Once PQB has accepted 2 electrons, it then grabs 2 protons, H plus day from the stroma to become fully reduced plastoquinol, or PQH2. And that removal of protons from the stroma is our first contribution to the proton gradient we're building. Then that PQH2 just diffuses freely within the membrane.
Okay, now for the other side of the reaction. Neutralizing that incredibly potent P680 plus day. This is where water splitting, or photolysis, actually happens.
As we established, P680 plus is the strongest biological oxidizing agent known to science. It has to pull electrons from water to prevent its own destructive decay. Now, since splitting two water molecules requires the simultaneous removal of 4 electrons to make one O2 molecule.
The PSII complex has to somehow accumulate 4 of those oxidizing equivalents one at a time.
Correct. This is known as the S-state hypothesis.
And what's the physical machinery that handles this really difficult 4-step accumulation?
It's a specialized structure. It's called the MN4CaO5 cluster, a manganese-calcium cluster. It's located on the luminal surface of one of the proteins, the D1 protein. It accumulates those 4 oxidizing equivalents, which are then transferred one by one back to the P680 plus via a very specific intermediate carrier. A tyrosine residue on that D1 protein, known as tears.
And why a tyrosine residue? What makes it good for that job?
Tyrosine, because of its structure, can easily switch between an oxidized free radical state and a reduced state. That makes it a really robust and essential intermediary. It's needed to stabilize the huge redox potential difference between the cluster and the P680 reaction center itself.
And the experimental proof for this S-state accumulation is visually pretty stunning. It's in figure 6.13.
It is. When researchers subjected algal cells to extremely brief flashes of light, they saw that oxygen production peaked reliably after every 4th flash. That confirmed that 4 separate photoreactions have to occur and accumulate before the structural change needed for O2 release can happen.
And crucially, the 4H plus produced by that water splitting are released directly into the lumen, which provides a second massive source for our proton gradient.
Right. But we do have to mention the Achilles heel of this powerful system, photoinhibition.
Operating at this level of power comes at a cost.
It does. High light intensity frequently damages the D1 polypeptide, the one that binds all the redox centers, and the M and COT cluster. So plants have to constantly and rapidly degrade and replace this specific damaged polypeptide through a continuous protein synthesis and degradation cycle just to keep PSII functional.
Okay, so with the electrons successfully boosted by PSII, the current now travels across the membrane bridge toward its second major power station, PSI. That PQH2 we generated diffuses through the membrane to the cytochrome B6F complex.
This complex is basically the molecular bridge connecting the two photosystems, and it acts as a proton pump, much like cytochrome BC1 in mitochondria. It oxidizes the mobile PQH2, which immediately releases its 2 protons, H plus O, into the lumen.
But the B6F complex is more than just an oxidizer. It's an active pump, which makes it a critical player in building that gradient.
It is. Using a mechanism known as the Q cycle, the cytochrome B6F complex is able to translocate 2 additional H plus from the stroma into the lumen for every pair of electrons that passes through it.
So for every electron pair, the cytochrome B6S complex contributes a total of 4H plus to the lumen. When you combine that with the 4H plus from splitting water, you can really see how the lumen quickly becomes highly acidic.
It does. After passing through cytochrome B6, the electrons are then transferred to another mobile carrier, plastothionein, PC. This is a small, water-soluble, copper-containing protein that operates exclusively on the luminal side of the membrane.
And plastocyan is the ferry. It carries the electrons over a short distance. The book says about 0.1 micrometers, since the 2 photosystems are spatially separated, to neutralize the PSI reaction center, P700 plus spec.
And now we've arrived at PSI. The second energy boost, focused on creating that vital reducing power, NADPH.
So light energy is captured by its own antenna system, LHCI, and passed to P700.
Right. The excited P700 transfers its electron to its primary acceptor, A0, which is another chlorophyll molecule.
And this creates the second charge separation. P700 plus QY, which is immediately neutralized by the plastocyanin that just arrived, and A0. And that A0 is an incredibly potent reducing agent. Its potential is about -1.0 volts.
We are now well past the energy level we need to reduce NADP plus quark.
The electron then follows this rapid channel of carriers.
It does. First pheloquinone, A1, and then through 3 iron-sulfur clusters, FX, FB, FA, which act collectively as an ultrafast internal molecular wire.
And that wire leads the electron to ferredoxin.
Ferredoxin is a small, water-soluble iron-sulfur protein that's associated with the stromal surface. It then hands the electrons off to the final enzyme, ferredoxin NADP plus reductase, which contains FAD, and that catalyzes the final reduction. NADP plus a proton plus 2 electrons gives you NADPH.
And the proton required for this reduction is taken from the stroma, which again contributes to that massive difference in proton concentration across the membrane.
So let's just consolidate the light reactions. For every 2 H2O and 2 NADP plus A, you get 1 O2 and 2 NADPH. And this whole process required 8 photons, 4 for each photosystem. And the key takeaway is that massive proton accumulation in the lumen driven by 3 distinct mechanisms.
Water splitting, the cytochrome B6FQ cycle, and the removal of H+ from the stroma for both NADPH and PQ reduction.
Before we move on to the payoff, we have to connect this elegant system to the real world, specifically the mechanism of some very effective herbicides.
It's a perfect illustration of how just disrupting one small step can crash the entire engine. Common herbicides like diuron and atrazine, they're targeted poisons that block electron transport right at PSII.
They work by binding directly to the QB site, and that prevents the binding and subsequent reduction of plastoquinone. No plastoquinol, no PQH2, means no electron transfer across that membrane bridge to PSI. It effectively just shuts down the entire electron flow.
And then there's paraquat, which targets the second photosystem.
Right. Paraquat competes with ferredoxin at PSI. When it intercepts electrons, it immediately transfers them to molecular oxygen, O2, and this generates highly reactive and destructive oxygen radicals. And these radicals then just attack and destroy the lipids and proteins inside the chloroplast, which leads to very rapid plant death.
So understanding the electron flow is essential to understanding how some of the most widely used agricultural chemicals actually work.
It is.
Okay, now let's move to section 6.5, photophosphorylation. The process of generating the chemical bank account, ATP, from that massive proton gradient we've built up.
The structure is almost a mirror image of the machinery you'd find in a mitochondrion. You have the CF1 headpiece, which is the catalytic site, projecting out into the stroma, and the CF0 base, which spans the thylakoid membrane.
And since all the protons accumulated in the lumen, the flow is from that highly acidic lumen-high H+ concentration back across the thylakoid membrane to the alkaline stroma, which has a low H+ concentration. And that flow drives the rotation of the ATP synthase turbine.
What's really fascinating here is the nature of the proton motive force, or delta P, in the chloroplast compared to the mitochondrium. In mitochondria, that force is primarily voltage-driven, but in chloroplasts, the force is dominated by the pH gradient, delta pH.
Yeah, the source material says the H+ concentration difference can be 1,000 to 2,000-fold. That translates to a delta pH of greater than 3 units. That's an enormous difference. It's far larger than what you'd typically see in mitochondria.
And that huge pH difference is maintained because the membrane allows neutralizing counterions to move freely, which prevents a large destabilizing membrane potential from building up. So that leaves the pH gradient as pretty much the sole driver of the ATP synthase.
The ATP we've discussed so far, the stuff derived from that straight-line flow of electrons from water to NADP plus REI, that's called non-cyclic photophosphorylation. But there's a second, equally important pathway that was discovered by Daniel Arnon back in the 1950s.
Right. Arnon observed that isolated chloroplasts could still produce ATP even if he took away the final electron acceptor, NADP plus zesiki. And this led to the discovery of cyclic photophosphorylation.
This pathway involves only PSI. It's completely independent of water splitting and PSII. So, how does it work?
Light excites P700, and the electron is transferred to ferredoxin, just like before. But instead of moving forward to NADP plus I, the electron is rerouted. It gets sent back through the cytochrome B6F complex and plastocyanin and returns directly to that P700 plus center.
So it's a closed loop, a short circuit that doesn't produce any NADPH or any O2. Why is this necessary? What's the point?
It's necessary to balance the books for the second stage of photosynthesis. The Calvin cycle, the sugar factory, needs an input ratio of 18 ATP to 12 NADPH to make 1 6-carbon sugar. Non-cyclic photophosphorylation just doesn't produce enough ATP relative to the reducing power it makes.
So cyclic photophosphorylation provides the necessary extra ATP.
Exactly. It provides the additional ATP that's required to regenerate the CO2 acceptor and just to meet the overall energy demands of the cell.
Okay, now that we have the power sources, the 18 ATP and the 12 NADPH, we can finally move into section 6.6, CO2 fixation and carbohydrate synthesis, the Calvin cycle, which happens entirely in the stroma.
This is based on the groundbreaking post-WWII work by Melvin Calvin, Andrew Benson, and their colleagues. They used the newly available radioactive carbon isotope, C14, and a technique called two-dimensional paper chromatography to map out the entire metabolic pathway.
And you can visualize the experiment. They exposed green algae to this radioactive CO2 for just a few seconds and then instantly quenched the reaction in hot alcohol.
When they ran the mixture on chromatography paper, they found that virtually all the radioactivity had accumulated in a single spot.
And that primary product was identified as 3-phosphoglycerate, or PGA, a 3-carbon molecule. This is why plants that use this pathway are historically called C3 plants.
The first logical guess was that the initial CO2 acceptor must be a 2-carbon molecule, but that wasn't the case. No. By tracing the radioactivity over slightly longer periods, they discovered that the initial acceptor is actually a 5-carbon compound, ribulose 1-bella-5-bis-phosphate, RoBP. The CO2 condenses with RoBP to form this unstable 6-carbon intermediate, which then immediately splits into 2 molecules of that 3-carbon PGA.
And the enzyme that catalyzes this pivotal reaction is maybe the most famous and most paradoxical enzyme in all of biology, Rubisco.
Ribulose bisphosphate carboxylase oxygenase. Its role is singular: convert inorganic carbon into the organic food supply for the entire planet. And yet, despite its importance, it is arguably the least efficient enzyme known. In terms of speed, its turnover number is agonizingly slow. It only fixes about 3 CO2 molecules per second.
So why has evolution settled on such a clumsy, slow piece of machinery for what is arguably the most important reaction on Earth?
It's an evolutionary trade-off. To compensate for its crushing inefficiency, Rubisco is produced in just staggering volumes. It is, by weight, the single most abundant protein on Earth. It can make up as much as half of the total protein mass in leaves. It has to be everywhere because it works so slowly.
So let's follow the full Calvin cycle, as shown in Figure 6.19b, through its 3 main phases in the stroma.
Right. Phase 1 is carboxylation. That's the fixation of CO2 onto RuBP, catalyzed by Rubisco. Phase 2 is reduction. The PGA is phosphorylated using ATP, and then it's reduced using NADPH, eventually forming glyceraldehyde 3-phosphate, or GAP.
And then Phase 3, regeneration, is the real energy bottleneck.
It is. Of the 12 GAP molecules that are produced, 10 of them are used in this complex metabolic rearrangement just to regenerate the initial 6 molecules of RuBP. And this regeneration step is highly energy intensive. It requires a substantial ATP input. 6 out of the total 18 ATP needed for the whole cycle are spent right here.
So recapping the net energy cost, to fix 6 molecules of CO2 and generate 2 net GAP molecules, the equivalent of 1 6-carbon sugar, the plant spends 12 NADTH and 18 ATP.
Which reinforces the absolute necessity of that cyclic photophosphorylation pathway we just talked about.
I mean, the whole system is shut down immediately in the dark. This light-dependent activation is managed by a really elegant system called redox control. It's in Figure 6.21.
So the electron flow from the light reactions doesn't just produce energy, it also acts as a regulatory signal.
That's right. When the light reactions are running, electrons that are transferred to ferredoxin get diverted to a small protein called thioredoxin. Reduced thioredoxin then acts as a biochemical switch. It converts these inactive disulfide bridges, SS bonds, on several key Calvin cycle enzymes into active sulfhydryl groups, SH.
And when the light turns off, the electron flow stops, the enzymes revert back to their inactive oxidized state, and the whole cycle just shuts down.
And we also have that pH mechanism.
Right. Running the light reactions pumps protons into the lumen, which makes the stroma alkaline. And many of the Calvin cycle enzymes are just naturally much more active at that alkaline pH. It ensures that carbon fixation only happens when the energy supply is guaranteed.
Okay, so once the primary sugar, GAP, is synthesized, what's its fate? Where does it go?
GAP has 2 main metabolic destinations. It can be exported out to the cytosol to synthesize sucrose, which is the primary transport sugar. It's carried through the plant's vascular system to fuel all the non-photosynthetic parts. Or it can remain right there inside the chloroplast to be converted into starch granules, which serves as a reserve food supply stored right there in the leaf cell.
Now we have to address the fundamental flaw in the system, the dark side of Rubisco, which is known as photorespiration.
This is the efficiency challenge that was born from evolution. Because Rubisco is a carboxylase oxygenase, under conditions of high oxygen and low carbon dioxide, it mistakenly attaches O2 to RoBP instead of CO2.
And the result of that is a chemical disaster for the plant.
It is. The reaction yields 1 molecule of 3-PGA, which is salvageable, and 1 molecule of 2-phosphoglycolate, a 2-carbon compound. And this phosphoglycolate initiates this energy-wasting series of reactions known as photorespiration, which can result in the loss of up to 50% of newly fixed carbon in hot, dry conditions.
The chapter details the complexity of this process. It's called the C2 cycle, and it involves this incredible cellular relay across 3 different organelles, which you can see in Figure 6.23.
Let's follow that wasteful carbon atom. The process starts in the chloroplast, which produces the phosphoglycolate and converts it into glycolate. That glycolate then has to be transported to the adjacent peroxisome, where it's oxidized to glyoxylate and then converted into the amino acid glycine.
And then that 2-carbon glycine molecule has to move from the peroxisome to the 3rd organelle in the chain, the mitochondrion.
Right. And in the mitochondrion, 2 molecules of glycine are combined to produce 1 molecule of the 3-carbon amino acid serine. And in the process, the plant releases that wasteful CO2. And although the serine can eventually be recycled back to the chloroplast, the net loss of carbon and the significant energy spent to transport all these metabolites and manage the pathway makes photorespiration a massive energetic drain.
And this inherent flaw exists because Rubisco evolved at a time when atmospheric O2 was practically non-existent.
Yeah.
But as the planet changed, plants had to adapt to combat this energy sink.
And that brings us to these fascinating evolutionary countermeasures, the C4 and CAM pathways. The fundamental problem for standard C3 plants in hot, dry conditions is water conservation. So they close their stomata,
Which dramatically lowers the internal CO2 concentration while at the same time increasing the internal O2 concentration. It's the perfect recipe for photorespiration.
It is. C4 plants, like tropical grasses, corn, and sugarcane, they solve this using spatial separation, which is shown in Figure 6.24. They have this unique leaf anatomy built around 2 concentric cell layers.
So the outer layer is the mesophyll cells. And here, CO2 is initially fixed by a completely different enzyme, PEP carboxylase.
And the crucial advantage of PP carboxylase is that it operates efficiently even at extremely low CO2 levels, and critically, it is not inhibited by O2.
So CO2 is fixed onto PP to form a 4-carbon acid oxaloacetate, or malate, which is then transported to that inner layer of cells.
That inner layer is the bundle-sheath cells, which are highly sealed off from the atmosphere. And once inside, that 4-carbon acid is quickly decarboxylated, which releases a massive concentrated burst of CO2.
And this internal release of CO2 within the bundle sheath can drive the local CO2 concentration up to 100 times higher than in the mesophyll.
That is the trick. This high CO2, low O2 environment completely saturates the Rubisco that's housed in those cells, forcing it to operate only in its efficient carboxylase mode. It virtually eliminates photorespiration, and the Calvin cycle proceeds efficiently within that bundle sheath.
But there's always a trade-off. C4 metabolism requires more ATP than C3 metabolism because the energy needed to regenerate that PP acceptor.
Exactly, and this is why C3 plants are still more efficient and dominant in cool, well-watered conditions where photorespiration isn't a major issue. C4 is an adaptation for hot, resource-limited environments.
And finally, we have the adaptation used by desert succulents, like cacti, sand plants. They use temporal separation.
Right. These plants fix CO2 using PP carboxylase only at night when the cooler temperatures allow them to open their stomata with minimal water loss. The resulting 4-carbon malate is then stored as malic acid in the large central vacuole, which actually gives the plant a sour taste in the morning.
And then, during the hot, dry day, when the stomata are sealed shut to conserve water, that stored malate is released and decarboxylated internally.
And this internal release provides a concentrated burst of CO2 for the Calvin cycle, which is run during the day using the ATP and NADPH generated by the daytime light reactions. CM plants essentially divide the 2 major stages of photosynthesis across the 24-hour cycle to conserve every last drop of water.
This has been an extraordinarily detailed journey through the engine of life, from that initial photon absorption all the way to the final sugar product. Let's synthesize the core takeaways.
Well, we charted the path from that ancient evolutionary challenge of splitting water, which necessitated the incredibly powerful P680 complex and the 2-stage Z scheme. We tracked 8 photons, converting light energy into the chemical potential of 18 ATP and 12 NADPH, using sophisticated molecular machines like the Q cycle to build that necessary pH gradient.
And we explored the stroma factory, analyzing the costly yet essential Rubisco enzyme driving the Calvin cycle, and then how plants develop these ingenious high-cost solutions, spatial C4 and temporal CAM separation, to mitigate the thermodynamic mistake of photorespiration.
It's just a beautifully complex system of coordination and dynamic regulation, making sure that the engine only runs when the light is present.
So we'll leave you with a final provocative thought, building on that paradox of Rubisco. We know Rubisco evolved when O2 levels were really low. However, Due to human activity, atmospheric CO2 levels are rising dramatically. We've moved from historic levels of about 270 parts per million to well over 390 parts per million today, and it's still climbing.
So given that a higher concentration of CO2 naturally suppresses that wasteful oxygenase activity of Rubisco, what profound impact might these rising CO2 levels have on the global competition between C3 crops, like wheat and rice, and C4 crops, like corn and sugarcane? The evidence suggests that mitigating the C3 plant's biggest weakness might inadvertently favor their efficiency in the future, which could fundamentally shift global crop productivity.
A truly fascinating possibility to explore as this global engine continues to adapt. Thank you for joining us on this deep dive into the chloroplast.
And thank you for joining us from the Last Minute Lecture team.